This is Fedra Pavlou, and I'm here with INNOVATORS-Kalaratne from Purdue University, and thank you very much for joining us today. Your research, which is the winner of the 2009 Young Innovators Award from the AAPS, focuses on the use of prostate-specific membrane antigen as a homing device for the imaging, diagnosis and the treatment of prostate cancer. So what problem are you trying to solve with your research?
Actually, if you look at current therapies for cancer and even other diseases such as inflammatory diseases involve treatment with cytotoxic drugs such as Taxol, vinblastine, methotrexate, and these drugs are distributed indiscriminately into all the body cells, thereby damaging to healthy cells as well as disease cells. So in the long run, this will lead to severe side effects. So what we're trying to do, we want to target these drugs to cancer cells or disease cells. So in that case, there are two ways you can do this. So one is by inhibiting enzyme or pathway that is uniquely required for the cancer cell or pathology of the cancer cell. One of the prominent example for this targeted therapeutic, they are called molecular targeted therapeutics, is Gleevec. So it's a tyrosine kinase inhibitor, and it inhibits it treats for chronic myelogenous leukemia. But at the same time, there's another way you can target drug, that is using a ligand that selectively target cell surface receptor on the cancer cells or disease cell. So by following this latter technology, we have developed targeted drugs for prostate cancer cells. So we are targeting prostate-specific membrane antigen that is overexpressed on the prostate cancer cells when compared to normal cells. So we have developed ligand that home into prostate cancer cells. So we are attaching drugs and imaging agents to this ligand, and so we have developed already targeted drugs for prostate cancer.
And what were the key results from your research?
So what we have developed from our research, so we have come up with using a crystal structure reported PSMA. So the crystal structure of PSMA is already available, and actually it was used to known as a glutamate carboxypeptidase 2. So we use this high resolution crystal structure, and we screen a library of compounds, for actual library of inhibitors, and then we have developed imaging agent, which is a radio imaging agent, and we have demonstrated using in silico. This is selectively bind to the PSMA, and then we have synthesized, and then we have demonstrated in animal models, xenograft link-caps tumor xenograft animal models, that our compound selectively accumulate in the PSMA positive link-cap tumors, and in contrast, when we pre-injected some excess of PSMA ligand or inhibitor, which is known as PMPA, so there was no uptake. So, and also we have demonstrated this does not go when you inject to PSMA negative tumor xenograft models. And from the biodistribution studies, we have demonstrated that our imaging agent selectively accumulated in the PSMA positive tumors, and also actually we have seen it is uptake in the kidneys, but when we pre-injected excess of PMPA, that is an inhibitor, well-known inhibitor, so both tissues showed that it won't accumulate our imaging agent, suggesting that these are PSMA mediated. But we have found that there's enough literature evidence to support that murine kidneys express higher levels of PSMA, but not human kidneys. So therefore, most of the other tissues did not express or shown any most of the radioactivity, so leading to very high tumor to tissue ratios. So when compared to currently prostacine is the current available FDA approved imaging agent. So when it was administered or tested in the animal models, link-up tumor animal models, the tumor to muscle ratio or tumor to background ratio was 3:1. But when compared to that, our compound is tumor to background ratio is 75:1. So you do the math, And then we have developed developed PSMA targeted optical imaging agent to demonstrate that our ligand can deliver multiple cargoes, and we were able to demonstrate that using flow cytometry and confocal microscopy. Our optical imaging agent binds into link-up tumors selectively, and with a couple of optical imaging agents, and also we were able to deliver siRNA to link-up tumor xenograft model, and also more importantly here, so the ability of these compounds to selectively label link-up cells motivate us to ask question: can we identify circulating tumor cells from the blood samples of the prostate cancer patient? So we conduct a double-blind study using 13 prostate cancer patients with metastatic disease and 18 healthy donors as a negative control, and our optical imaging agent DuPA-FITC was able to identify circulating tumor cells from the prostate cancer patients' blood samples, but we didn't see any fluorescently labeled cells from healthy donors. And with this, all the background with our hand, for selective delivery of radio imaging agents and then optical imaging agents, encouraged us to develop PSMA targeted chemotherapeutic agents. So in that case, we have a screen library of conventional chemotherapeutic agents against link-up cells, and we were able to identify many potent drugs that can kill the link-up tumors, so tumor cells. And then, but these are, as I told you, these are non-targeted. So therefore, we were able to targetedly deliver these drugs. So I will give one example, that is PSMA targeted tubulizine B. So tubulizine B is a microtubule inhibitor, and it is unfortunately this drug kills the healthy cells and normal and cancer cells, leading to severe side effects. So that inhibit or hindered this being used in the clinic. So what we did, we attached this drug to our ligand via releasable disulfide linker. So once this whole compound binds to PSMA, it's a cell surface antigen. So once it internalizes through clathrin-coated bits inside the endosomes, this disulfide bonds break down, and then it will release this our drug, original drug, and we were able to demonstrate that our drug kills link-up tumor cells with the IC50 of 3 nanomolar, and also we were able to demonstrate in tumor xenograft models that our drug cures the established link-up tumor xenograft within 12 days, and non-treated mice tumor grows over 1000 millimeter cube within that period. And also when we inject excess of PMPA, that is an inhibitor of PSMA, and then after that injecting our compound, did not cure the tumor. So that shows our compound is targeted. And more importantly, during this study, weight of the mice did not change, so that means there's no toxicity. And from according to pathologists, so there's no toxicity was observed in most of the tissues, including bone marrow, and there were minor toxicities like enlargement of kidney and liver and spleen was observed, but according to our pathologists, those are negligible. So what this boils down to, by targeting these non-specific and highly toxic chemotherapeutic agents, we can eliminate the side effects. So that's the outcome.
Pretty incredible stuff, especially with the, I suppose, the length of time that the drug actually took to kill the tumour cells, which you state to be around 12 days.
Yeah, 14 days.
If you were to consider its use in a prostate cancer patient, how would you believe that therapy would be administered once the correct dose regimen was discovered? What would be the hope that perhaps this therapy would kill existing tumour cells, but would it ever be used continuously just to prevent tumours in prostate cancer patients above and beyond, say, 2 weeks, for example?
So actually this is just a pilot study, proof of concept study. So let me go back a little bit. So our imaging agent, radio imaging agent, we are starting clinical studies in this December, and our optical imaging agent already in the clinic to identify circulating tumor cells from prostate cancer patients' blood sample. So therefore we can screen patient for that. So these two imaging agents, we can tell which patients will respond to our therapeutic agent. So with this data in our hands, so once we do this imaging agent, once it tests with the patients, so we will know where it goes other than tumor. So with that data in our hand, we will take our drug to clinic. So most probably, so we have to inject IV. So and basically, you have to give the chemo, like this is targeted chemo. So and then after that, you have to follow up.
But you hopefully don't believe that the side effects with this therapy would be comparable to the side effects of other chemotherapeutic agents. So hopefully those severe side effects of chemotherapy should hopefully not apply to this agent if it went into clinic.
Yes, one of the major side effects going to be bone marrow toxicity. But we have shown in animal models that there's no bone marrow toxicity. And so we do not expect any side effects, but at least we'll minimize the side effects. So, but at the same time, I want to go to a new concept. That's, there's a hypothesis that even though you cure cancer, so there's most of the cancers relapse or recur because the existence of cancer stem cells. So, therefore, until we cure cancer stem cells or kill the cancer stem cells, we may not be able to cure cancer. So my next goal is to develop targeted drugs for cancer stem cells. So I'm already lined up with my postdoctoral studies with Scripps Research Institute. So over there I'm going to develop targeted drugs for cancer stem cells. And so it will kill the stem cells plus bulk of the tumor.
Well, that's wonderful. So you've already explained the next area that you're looking into, which is fantastic, and I wish you every success with it because obviously that is the main goal. And I don't know if any other researchers are looking into killing cancer stem cells at the moment or how they have progressed. Do you know if there's any data out there at the moment that's been published into that area?
Actually, right now, currently people, there's a huge development in this area and a lot of research going on to kill cancer stem cells, and a couple of articles has been published in Cell and Nature. Kind of people are screening library of compounds for cancer stem cells, and some of the people try to kill using the signal transduction pathways like P53, P10, AKT, and kind of pathways, and stop the mutations kind of things. And even those drugs, those drugs are not targeted, as I told you. So it will affect the same way to other cells also. So my idea is we will find the cell surface receptor or antigen or biomarker. So I don't want to give my idea right now, but there are strong evidence that cancer stem cells can generate new cells that has mutations, or they are resistant to all drugs. And next thing is, when cancer metastasizes, it won't go to all the parts of the body. They selectively go to some organs. So what is reported currently, so tissues that release SDF1, that means stromal derived factor 1, most of the target for cancer metastasis, because cancer stem cells has receptor called CXCR4, that is a chemokine receptor, and it's the cognate receptor for SDF1. So therefore this cancer stem cells goes wherever this SDF1 release, and then homing there and start metastasizing the cancer. So my target is to develop drugs by targeting CXCR4.
Thank you very much for explaining that to us, and also thank you for joining us today and for discussing your research into prostate cancer as well as your future targets as well. So I'd just like to say thank you very much and congratulations on the award as well. And just to remind listeners that they can actually download your full presentation that was a winner of the award on our website, farmtech.com.